Hello, and welcome to our lecture on the cell cycle, is principal characteristics of the cell cycle, and chromosome packaging. We're going to talk a little bit about how chromosomes are packaged as well. So let's dive on in. So first off, chromosomes. Chromosomes are how we package here. Eukaryotic organisms package their genetic information. As humans, we have 23 pairs of chromosomes. We are a diploid organism. As diploid organisms, we are referred to as 2N. This is because we inherit one chromosome, one of each chromosome from our mother, that's maternal inheritance, and one of each chromosome from our father, that's paternal inheritance. So haploid organisms are 1N and diploid organisms are 2N. Our gametes, sperm and eggs, are 1N organisms, and so when the two haploids come together, we create a 2N organism. Of our chromosomes, we have 23 pairs. 22 are considered homologous. One pair is considered non-homologous, but this only is true if you are a male. And that's because our 23rd set of chromosomes are the sex chromosomes. So all of our chromosomes are in pairs. We have a total of 46. 44 of them are homologous, so we've 22 sets of homologous chromosomes, and then one pair of sex chromosomes. Again, females have 2 X chromosomes, so they are homologous in all sets. Males are XY, which are non-homologous chromosomes, so they do not have 23 homologous pairs. In cytogenetics is a field that falls under the umbrella of genetics that observes and studies chromosome structure, packaging, and anomalies in chromosomes. So let's take a little bit of a look here at the field of cytogenetics. A lot of genetic counselors will utilize cytogenetics when they are counseling couples that may have genetically inherited diseases. So in cytogenetics, we can see where our chromosomes pair up. And how do we look at what's known as a karyotype? You may have actually completed a karyotype in lab. I know that's one of the exercises you do in lab. And a karyotype is created by first creating a chromosome array. So they will take a blood sample. The blood cells are centrifuged so that we get a nice pellet of blood cells in the bottom of our tube. The supernatant or serum from the blood sample will then be removed. That blood pellet is then resuspended in a hypotonic solution, and this is so that these cells will lyse open, and any white blood cells that may be in the sample will then release their genetic information in the form of a chromosome. The sample is then centrifuged again in order to get all of the blood cell membranes in that down at the bottom, and this will then be removed. The cells are suspended. The supernatant is removed. The cell pellet is then suspended because the chromosomes are going to be down in these cell pellets, is suspended in a type of fixative solution and then placed on a slide. When we look at these under the microscope, I just want you to see right here is kind of what you see in a chromosome array. Now here it just looks like kind of a big mess, but photographs will be taken of these, and there's computer software that will sort these chromosomes based on their staining pattern, how they were stained when they were on the slide here, the staining pattern that occurs, as well as the length, size, and shape of those chromosomes. So here we have a karyotype. This is for a 2N organism. This is a human karyotype, and we can tell that it's from a male because we have the X and Y chromosomes, which is necessary for formation of a male. On the left here, we have another type of karyotype. Now, these were with fluorescent microscopy, whereas these were probably what's called an eosin or hemolysine stain. Just a different type of staining method. But the banding patterns are what we look at. So this is fluorescent microscopy over here on the left. And you can see we have these same banding patterns. These are very important for matching up these chromosomes. They have the same colors. And the different colors are coinciding with different densities and regions of DNA. So all of these banding patterns definitely help with recognizing which sets of chromosomes from this large kind of array over here, how do we match them up. Now, this matching of chromosomes here, this looks like a pretty healthy individual. If we have things like muscular dystrophy or a condition called trisomy, in trisomy, we may have a chromosome set, instead of having 2, we have 3. And if we have 3 of a set of chromosomes, in this case, we can have a disease state. So that's something like muscular dystrophy. Down syndrome is another one, is a trisomy disease. So that brings us to chromosome packaging. We've already looked at how DNA is replicated, but the replication of DNA is done in order to prepare the cell for cellular division. And in order for the cell to divide after DNA replication, the DNA has to be condensed and packaged so that cellular division can occur, which is part of the cell cycle that we'll look at next. So that means we need to look at the stages of chromosome packaging. DNA, we have already looked at when we looked at gene expression, genetic regulation, DNA replication, transcription, translation. We looked at all of that as DNA in its normal double helix type form. Uncondensed, accessible, just a big long double helix like you can see here on the left of DNA. So that is stage 1. And in this case, the DNA is not packaged or anything. It's just in its double helix form and it's functional. But DNA begins to, once it's replicated, the first thing it's going to do is begin wrapping around a set of proteins called histones. Histones are octamer proteins. There are 8 actual polypeptide chains that make up the quaternary structure of a histone molecule. The histone itself, histones 2 through 8 are found in the center here, composed of, they create this kind of big wheel-like structure, and the DNA is wrapped around the histone about 1 3/4 to 2 times. It is anchored to the histone to the histone complex by a histone known as H1. H1 is also referred to as the linker protein or linker histone. And this actually anchors, you can see it kind of holds the DNA wrapped around the histone here. When I have DNA wrapped around a histone complex, this is referred to as a nucleosome structure. And these nucleosomes will zigzag, create a zigzag-like structure. And when they create this zigzag-like structure from the width of this, you can see because of how these guys anchor and where they anchor, these nucleosomes will alternate back and forth. So in the nucleosome structure, it creates what's referred to as a 30 nanometer fiber. And that 30 nanometers comes from, comes from the width of the fiber. How wide across is it from here to here? Well, it's 30 nanometers. So this all measures 30 nanometers. That's why it's referred to as a 30 nanometer fiber. Once the 30 nanometer fiber is created, the DNA begins to kind of zigzag again and create what's called radial loop domains. The radial loop domains are anchored to the actual nuclear matrix. There's this right here. I'm going to show you this micrograph right here. This is an electron micrograph of our radial loop domains. And this is a bunch of protein here known as the nuclear matrix. And that nuclear matrix, we studied that when we looked at cellular structure. The nuclear matrix will be used to anchor these radial loop domains and cause these radial loop domains to begin compacting even further. So our 30 nanometer fiber moves into radial loop domains. That's what we see here. And the radial loop domains will begin to supercoil or compact on each other to create what's called heterochromatin. Heterochromatin is condensed DNA. And when it's completely anchored and forms these tetrads, it'll create the scaffold from that nuclear matrix. And once it's bound to the nuclear matrix itself, that scaffold, we have our complete formation of chromatin. So this is our 30 nanometer fiber formed into radial loop domains to create this condensed form of DNA known as heterochromatin. And then when heterochromatin attaches to the nuclear matrix, we have our true chromatin. So this is heterochromatin attached to matrix. Now that brings us to the concept of heterochromatin versus euchromatin. Now, heterochromatin is that highly condensed form of genetic information, or DNA, that is not accessible. That's really what's happening here, is we have packaged the DNA so tightly, it's so supercoiled onto itself and wrapped around those histones and so densely packaged that we have no way of getting in here to get a transcription factor or an inhibitor, a repressor, or an activator. None of those types of proteins that are associated with genetic expression that we looked at in the last unit are able to access the DNA in here. It's very tightly wound up onto these nucleosomes and creates those radial domains that kind of attach to each other, and then they get attached to the nuclear matrix, and it creates this really dense form of genetic information. So heterochromatin is inaccessible, which causes it to be non-transcribed. So any genes that are wrapped and tied up in the DNA that's in this really condensed form of heterochromatin, anywhere we see heterochromatin, there it is non-transcriptional. Those genes are not being used. There are some regions of the DNA, however, that will always remain in a euchromatin state. So there may be packaging and such, but there will be a region of DNA that will be in a euchromatin state, which is a transcriptional state. So here, in order to make the DNA accessible and loosen things up, instead of modifying the actual DNA itself, we modify the histones. Remember, histones are proteins, so if we attach something to a protein or remove something from a protein, that protein is going to conformationally change. And that change in conformation can cause those histones, right? We're going to modify the histones, causing conformational changes in the histones that are going to loosen up that packaging. If we loosen up that region of the DNA, we can then move in there as a transcription factor, activator, enhancer, silencer, whatever it may be, and we can move in here and then start actually transcribing those genes. So these little stars on the black circles here are regions or areas on histones that are being modified. And these modifications occur through either methylation, adding CH3 groups, acetylation, or adding free phosphates through phosphorylation. Remember, you attach something to a protein, it's going to change the shape of the protein. So where on that histone we add something or attach something in order to activate a particular gene is going to be, of course, gene specific. But euchromatin is transcriptional DNA. Heterochromatin is densely inaccessible, non-transcriptional DNA. We can have regions even after replication, if we're in it, like what's referred to as the G1 phase of the cell cycle, where the cell is kind of doing its normal everyday type of thing. There are still regions of DNA that will remain in a heterochromatin state, and the only time they ever exit a heterochromatin state is for DNA replication. Otherwise, they will always remain in a heterochromatin state, and that is because the genes associated with that region of that chromosome are not to be transcribed. They may have been used during embryonic development, or they may be used in very, very specific special circumstances, or they may never be used by that particular cell type at all. Such as if you're a kidney cell, then regions of your chromosome that would make you a cardiac cell are going to remain and forever remain in a heterochromatin state, except for DNA replication. So that brings us to the cell cycle. And the cell cycle is really just kind of the life of the cell. How long is this cell going to be around? And our cells, right, must divide. And cellular division occurs in order to repair broken cells, replace cells that have died, and of course to instill growth for multicellular organisms. We started out as one cell, but as we grow and get larger and increase in size, a lot of that is, of course, that is due to cell division and an increase in the number of cells. And we know from the cell theory that all cells come from pre-existing cells. So what's really happening here in cell division? Well, cell division in the cell cycle is divided into actually 5 different phases. There's 4 general, but the 4th one is really two phases in one. So we start out in the G1 phase. And in the G1 phase, as far as mitosis is concerned, we're in interphase, and we will cover mitosis in our next lecture. So in the G1 phase, this is one of the longest phases of the cell cycle. This is where cell growth is occurring. And what we mean by that in G1 is we have transcription and translation. We're producing proteins. Cells are carrying out their normal function. And the cell is also getting ready to prepare for cellular division. From G1, we move into the S phase. The S phase is primarily the replication of our genetic material. DNA is being replicated inside of the nucleus in order to prepare for division. From the S phase, we move into G2. In G2, cell growth is occurring, but this cell growth is really occurring simply through expression of proteins associated with mitosis. So we're going to have to make a lot of microtubules and kinetochore proteins. All of those proteins, structural and non-structural, that are associated with a cell dividing are going to be produced in G2. And then at the end of G2, we move into the mitotic phase or the M phase. This is referred to as the M phase. In the M phase, mitosis occurs. And right after mitosis, within that M phase, is the final part of cell division known as cytokinesis. At the end of the M phase, which includes both mitosis and cytokinesis, we will have the formation of two daughter cells. Each of those daughter cells then move into their own G1 phase and repeat the cell cycle again. So what's really happening in each of these phases? So in the G1 phase, again, this is cell growth, and the cell is metabolically active. It replicates any organelles, right? So towards the end, this is about a 10-hour phase, sometimes 12 hours, depends on the cell. But in G1, we're going to move through G1. As we move through G1, different proteins associated with cell division and cell cycle are going to accumulate in concentration. And as they begin to accumulate in concentration, this is going to signal the cell that it's preparing to move into the next phase of the cell cycle to prepare for cell division. If that's going to occur, then we're going to start seeing duplication of organelles. Other parts of the cytosol are going to replicate. The centrosome begins to replicate because the centrosome is responsible for microtubules and such, and structurally those are going to be required for the cell to actually create two new cells. So at the end of G1, when we get towards the end here at G1, the cell is going to have to make a decision, and that decision is going to be protein determined. If we have an accumulation of a protein known as G1 cyclin, when that protein increases in a high enough concentration, the cell will move through a checkpoint to move into the S phase of the cell cycle. But if G1 cyclin is not produced, if it's repressed or silenced, the genes for that are silenced, then the cell will, instead of moving into S phase and moving into the next phase of the cell cycle, the cell will actually enter into senescence. And this is referred to as G sub 0. Senescent cells are cells that do not replicate, but they don't die either. They just simply carry out their function without cell division. Senescent cells include things like cardiac cells and neurons. These are cells that if you destroy or damage these cells, you do not make new ones. And that is because these cells are in a G sub 0 or senescent phase. They are non-dividing. So if our cells don't divide, they cannot repair, they cannot replace, and they cannot increase through cell growth. Once we get into get to the end of G1, and let's say that our G1 cyclin protein concentration is high enough, the cell now kicks over into S phase. In the S phase, we have DNA replication. The genetic material will be replicated and then packaged and condensed up like we saw in chromosome packaging just a few minutes ago, and the cell then enters into G2. In the G2 phase, the cell does continue to grow, but we get further compaction of the genetic material, and we start to see synthesis of proteins, more proteins associated with chromosome replication. And this will include things like kinetochores and microtubules, nuclear matrix, those types of proteins that are associated with that final packaging phase, as well as movement into mitosis. At the end of G2, we move into, we go through a checkpoint to move into the mitotic phase. In the mitotic phase, mitosis finally occurs. And in mitosis, we're going to study that in the next lecture. We go through all the phases of mitosis. And at the end of mitosis, and before entering into G1, we move into cytokinesis. So mitosis is division of the nucleus. And the nucleus divides first, and once nuclear division has occurred, because our nucleus contains our genetic material, once nuclear division and division of the DNA copies is completed, the cell then moves into cytokinesis. Cytokinesis is where those organelles that were replicated now divide into each half of the cell, and the cell membrane will pinch off and we create two new cells. So at the end of the M phase, we go from one cell to, of course, two. And then each of these cells will then enter into their own G1, and the cell cycle will begin again. So advancement, how do we advance from one phase of the cell cycle to the next? Because some of those advances are seamless and others are actually stopped. So we have what are called checkpoints, and these checkpoints occur at the end of G1, the end of G2, and the middle of the M phase. And what really happens in the M phase is we go through mitosis, and between mitosis and cytokinesis will be a checkpoint. And these checkpoints are manned by a set of proteins called CDKs. CDK stands for cyclin-dependent kinase. In addition to the CDK, we also have a set of proteins called cyclin proteins. And all of these, they're called cyclins because this is known as the cell cycle. So they are associated with cyclic structures. All right, as we move through, let's start with our G1 here. So as the cell is moving through G1, we're going to assume non-senescence here and that we're advancing through our cell cycle as normal. As the cell moves through G1, it begins producing a protein known as G1 cyclin. And this G1 cyclin begins to bind to the cyclin-dependent kinase, or the G1 CDK. And these two proteins together create the G1 cyclin CDK complex. This complex becomes active as a kinase. So it has kinase activity. When it works as a kinase, the kinases phosphorylate. Right? So this CDK complex here is now going to reach out to other proteins that are associated with DNA replication and begin activating those proteins associated with DNA replication and with running the S phase. When this concentration gets high enough, then we can pass this checkpoint and move into that S phase. Once the cell moves into the S phase, after it enters the S phase, the G1 cyclin will be degraded and broken down so that this complex will fall apart, G1 is degraded, and the cell will no longer be producing any of it because it's in DNA replication. The CDK, however, will remain behind, so we still have CDK present. As the cell moves through the S phase and into the G2 phase, during this, a second cyclin is produced called a mitotic cyclin. The mitotic cyclin will do the same thing. It will begin binding to the CDK, and as it binds to the CDK, it begins phosphorylating now proteins. The mitotic cyclin complex will begin phosphorylating proteins associated with mitosis, such as producing kinetochore microtubules and astral microtubules, nuclear matrix, those sorts of things to help complete that DNA replication. In addition to this, DNA has been replicated, so both within the S phase as well as in the G2 phase, we're also looking for DNA damage. So we're looking to make sure and double checking, so things like DNA polymerase 1 and DNA polymerase 2, which are part of the repair mechanisms in DNA damage, will also be active, and they're going to be checking that genetic material prior to compaction to make sure that everything was copied properly. Once we reach that threshold of CDK mitotic complex here, this kinase, we kick into the M phase and mitosis occurs. In mitosis, we have separation, or we have a division of the nucleus. At the end of nuclear division and prior to cytokinesis, we have the metaphase checkpoint. And in the metaphase checkpoint, it's simply a set of proteins that are going to make sure that chromosomal division during mitosis occurred properly. And we're going to look at how chromosomes separate in mitosis in the next lecture. Once we get through the mitotic phase, this checkpoint is passed, cytokinesis occurs, there's no turning back. We now have two completely separate cells, and each of those cells will then repeat into their own cell cycle from there.
Cell growth and division are divided into phases. Cells grow during the G1 phase, and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclin starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again. Cell growth and division.
So some things I'd like you to think about from this particular lecture, some questions that you should address to prepare for the exam, at least for the portion of the exam on cell cycle and chromosome packaging. So what are the different levels of chromosome packaging and what's happening in each of those levels? What is the difference between euchromatin and heterochromatin? What are radial loop domains, and why are these radial loop domains important to chromosome packaging? What is the purpose of the cell cycle? What are the phases of the cell cycle and the characteristics of each phase? What's happening in each of those phases of the cell cycle? What is the cell doing? What are the cell cycle checkpoints and how are those checkpoints regulated? How does a cell move from one part of, from like a G1 to an S phase or S to a G2? How does the cell advance from one part of the cell cycle to the next? What are cyclin-dependent kinases or CDKs? How do they work? How does a cyclin-dependent kinase regulate the cell cycle? Where do those checkpoints occur? And think about the cell cycle as a whole and how the regulation of a cell moving through the cell cycle may be associated with things like different types of cancers. What would happen in a cell if a checkpoint wasn't working? Or how does a cancer cell utilize, as a cancer cell, how could a cancer cell utilize or use the cell cycle to its advantage? What could it do to the cell to change how the cell cycle affects movement and cellular division? So those are just some questions I want you to think about and use to help you prepare for the exam. In our next lecture, we will talk about mitosis. After mitosis, we'll talk about meiosis, which is formation of gametes, and we'll compare those two phases together. Have a good day, and I'll see you in the next one.